Hi everyone, my name is Kayla Johnson and I'm a PhD candidate at Michigan State University. Today I'm going to talk about my recently finished work titled Robust Normalization and Transformation Techniques for Constructing Gene Co-expression Networks from RNA-seq Data. Currently there is a lot of gene expression data publicly available, and of course we only expect this to continue growing. This is great for those of us interested in building co-expression networks, as they are excellent tools for many applications including module finding, gene function prediction, and gene disease prioritization. However, creating these networks is not straightforward because we need to process and normalize the data, which can have large impacts on the final network. There are a number of methods that have been developed for normalizing and processing RNA-seq data, but it's not clear what methods in which combination create the most accurate co-expression network. The most extensive work in this area is part of a 2015 study which did a lot of excellent work, but the focus was quite broad, so there's still room for improvement in regards to normalization comparison. In our group, we have a lot of interest in network integration methods and applications of aggregated networks, which means we are interested in finding robust methods that consistently yield the most accurate co-expression networks. Improvements I'm specifically interested in here are increasing the amount of data to compare methods across a large number of diverse data sets. I wanted to test more normalization methods and further the combinations of methods. In the original work, one standard pipeline was chosen and steps were swapped out one at a time, but since we can use multiple methods, I'm interested in how combinations impact network performance. I also wanted an evaluation based only on the accuracy of edges in the resulting network, as I want the network to be maximally useful regardless of downstream task, rather than evaluating performance on a specific prediction task. And lastly, I chose a precision recall curve over AEROC as the main evaluation metric because in cases where you have far more negative than positive examples in your gold standard, such as the case with co-expression analysis, the precision recall curve is more informative than AEROC. As I said earlier, I really wanted to compare method performance across a lot of diverse datasets. For that, I turned to the Recount2 database and downloaded any experiment from the Sequence Read Archive with at least 5 samples from a tissue that has at least 5 representative datasets. This turned out to be over 6,000 samples from 20 different tissues. In addition to this data corpus from many different labs, I also downloaded available GTEx datasets for robust comparisons on a large, high-quality, more homogeneous corpus. In total, I collected almost 16,000 samples from 38 tissues. To build each co-expression network, I started with gene counts from Recount2, then did some filtering for sparse samples and genes, aiming to keep as much data as possible. I tested four methods for within-sample normalization, which is a type of normalization designed to allow fair comparisons of read counts between different genes within a single sample. And 4 more options for between-sample normalization, which is meant to make the same gene count in different samples more comparable. Only protein-coding antisense RNA or lincRNA genes were included in my network, and I used Pearson correlation to calculate edge weights. Network transformation is a technique that adjusts edge weights based on the topology of the network, and I tested 3 options here. Finally, I made both tissue-naive and tissue-specific gold standards to evaluate the ability of networks to recapitulate generic and tissue-specific gene functional relationships. I started with a set of manually selected gene ontology biological process terms that were deemed specific enough to have confidence that any pair of genes co-annotated to that term could be considered functionally related and a positive edge in the gold standard. We ignored gene pairs that were not co-annotated to a specific term if they interacted with enough of the same genes or shared an annotation to another set of manually selected general terms. Then we set the rest of the edges as negative. Tissue-specific standards were created by subsetting the naive gold standard based on genes known to be expressed in that tissue. Altogether, there are 30 possible combinations of methods, which I'm going to call workflows. And here I'm showing you the overall performance of our 30 different workflows in the GTEx data evaluated on the naive gold standard. TMM and upper quartile normalization tends to be at the top, and CLR also seems to work quite well. Within sample normalization performance tends to vary more with other choices in the workflow. We see a lot of similar trends in the SRA data, though we can see that overall GTEx networks tend to be more accurate than SRA networks. This result confirmed my suspicion that workflows can yield very different results. So I wanted to determine the significance between methods. In this heat map, a red box indicates that the workflow in the row outperformed the column workflow on more datasets. I've added a star for significant comparisons based on a paired Wilcoxon rank sum test. The bar plot shows how many times a workflow was significantly better than another workflow in the GTEx data and in the SRA data, where we confirm that the top workflows are in fact significantly better than most other workflows. From this, it is still unclear how much an individual method choice impacts a workflow. So, to assess how including a particular method in any given workflow affects general performance, we aggregated all workflows which include a particular method and calculated the proportion of times that including that method resulted in the workflow being significantly better than one that does not include that method. Both GTEx and SRA data agree that not using within sample normalization and using TMM or upper quartile normalization are the best choices in their respective groups. But CLR tends to work better than no network transformation in GTEx data, while the opposite is true for SRA. GTEx data and SRA data differ in many respects, but the most obvious was sample size. So, using the large GTEx datasets, I randomly sampled each GTEx tissue for a total of 10 trials for each of 9 representative sample sizes. SRA datasets also vary considerably in terms of other factors, including sample similarity, number of mapped reads, and tissue type. So the GTEx resampling networks were used to investigate effects of each of these factors. To be practically beneficial, the best workflow for constructing co-expression networks should result in consistently high performance irrespective of variations in these experimental factors. I do not have time to talk about all the details of this analysis, so I'll just say that it confirmed the robustness of our top workflows. I've just thrown a lot of information at you, so to sum up, I built 81,600 networks, so you don't have to, and I can now give you these recommendations. First, if you can get gene counts, always use TMM or upper quartile normalization. TMM tends to slightly outperform upper quartile. If your data is only available already normalized, TPM consistently outperforms CPM and RPKM, and you can convert CPM and RPKM to TPM. If you are lucky enough to have over 40 samples, you should use CLR to transform the network. And last, quantile normalization and WTO transformation should both never be used, as in all circumstances, they always hurt performance. And I'd just like to quickly thank my awesome lab for their continued guidance and support before I open it up for questions.